Really appreciate the opportunity to speak today and get a little extra time. Um, I'm going to save sort of the journey, self-discovery side till the end of my talk, and really get right into the science first. So I will caveat all of this with I've been at Scripps for quite a long time, and I was a graduate student, a postdoc, and a staff scientist, and now I'm a senior staff, but I'm also a um, principal investigator on my own R01, and I'll talk a little bit about that at the end, which is this new fantastic opportunity for early stage investigators to get into their own area of science without having to rely on years and years of experience and preliminary data. But sort of where I've been in the past several years is studying infectious disease. And this is sort of a hallmark of what we do at Scripps. And in particular, I'm involved in the structural biology of how the immune system interacts with viruses. And today I'm going to primarily talk about HIV, but a lot of the stuff that I am working on and that I have worked on is broadly applicable to pretty much any virus. And so what we've done in the lab that I work in, which is Dr. Andrew Ward's lab, is basically look at how viral glycoproteins from viruses like coronaviruses, HIV, and Ebola interact with antibodies from either vaccinated animals, vaccinated people, or in the case of HIV, how people that have HIV and are elite controllers, so they don't really need medicines to control their infections. So we're very curious how these people are able to do that and how these vaccines are working. And the way we do that is by combining antibodies with soluble versions of these glycoproteins, generally speaking, and then using cryo-EM to solve structures of the antibodies bound, and then subsequently mapping the immunogenic landscape of these antibodies and trying to gain an insight into what type of immune response results in protection. So the other side of that is these antibodies generally are thought to neutralize these viruses. And so that means they directly block them from entering cells. There's many different mechanisms behind how they actually do that. But, to long story short, generally speaking, neutralizing antibodies are thought to be the primary types of antibodies that you want to have to prevent viral infection, especially from HIV. But the other side of the spectrum here with these antibodies is that they don't only neutralize viruses, they also have this ability to alert your own immune system to the presence of a viral pathogen. And this is done generally when a virus enters a cell and it starts to replicate. And then prior to budding, a lot of these glycoproteins and viral proteins are exhibited on the surface of these cells. And antibodies that you might have already circulating in your system, or that can be given, for example, as like a therapeutic, would bind to these cells and alert your immune system to the presence of these viruses. And these immune cells could then attack the infected cells and prevent them from spreading virus. So I am in particularly interested in a particular type of cell called a natural killer cell. And these have been implicated strongly in people that control HIV, as well as some early vaccine studies that have been done as being strongly correlated with protection from viral spread and infection. And it's generally referred to what these antibodies do with these NK cells as ADCC, or antibody-dependent cellular cytotoxicity. But the thing that we found is that these antibodies bind in many different places on the virus, and I'll show you that in just a second. But they are very variable in their ability to perform ADCC. So basically what you're seeing here is a study done by another group, and these are two different viruses, one on the left and one on the right, different strains. And in blue are broadly neutralizing antibodies that, or excuse me, in blue are are non-neutralizing antibodies that bind to these pretty common sort of what we call epitopes. And then in red would be the ones that are broadly neutralizing. And sort of what you can see is that depending on where these antibodies bind, some of them are really good at ADCC and some of them are pretty crappy at ADCC. But there's not really a defined pattern here. And in our lab and in others, we have mapped these antibodies and many, many more beyond the ones that are known to the surface of the viral glycoprotein for HIV, which is referred to as OMV. And what we find is that these antibodies combine over a very broad landscape. So although there are these common sites of vulnerability that you see on the right that we've identified, actually these antibodies bind at these widely varied angles of approach. And so I and others had thought many years ago that potentially this might have something to do with this varied ability to perform ADCC. And so what kind of led me into thinking that was this paper that came out just a few years back. And actually my previous graduate advisor, Dr. Eric Allman-Sapphire, solved one of the first human antibody structures, which happened to be an HIV antibody, B12, as a matter of fact. But in the crystal structure packing, they noticed these interesting hexamers that the FC domains, and the FC domain is the portion of the antibody that would interact with cell surface receptors on your white blood cells, for example, NK cells. And I'll tell you a little bit about that in a moment. But it turned out in the crystal packing that these FC domains were packing together and interacting with each other. And so it was explored further, and it turned out basically that this was not a fortuitous type of crystal packing event, that indeed the contacts made here were extremely important for complement deposition. And if you interrupted that interaction, that complement in vitro assays went way down. And then even more recently, there was a paper that came out in the cancer kind of environment that implemented the angle of approach of an antibody with differential complement deposition and activity. So these scientists solved a few structures of this antibody called rituximab. And then there's a couple of different versions of rituximab, OFA and OBZ, and on the far left, you can see those structures. And basically they bind to CD20 and they are anti-cancer drugs, but some of them work really well and some of them don't work so well. So it turns out that rituximab binds at a really shallow angle, and it's thought, and it also binds two Fab domains, or so these are sort of the arms of the antibody per CD20 molecule. And this antibody performs CDC quite well. There's a different version that binds much more steeply, and it turns out that it performs complement a little bit less well, but still okay. And then there's a third version that they refer to as like a type 2 version of rituximab, and it doesn't perform complement very well at all. As a matter of fact, it only binds one antibody per CD20 molecule, and so its angle of approach is widely altered. And so we developed this theory that depending on the angle of approach that an antibody makes, the FC domain of that antibody might be more or less accessible by these receptors on effector cells, and in this case, NK cells. And so I had this working hypothesis that if we could take these antibodies and classify them based off of their angle of approach, and then look at their ADCC activity, that we might be able to correlate that with sort of what I'm calling immune complex geometry. So a little bit more about these receptors that I've been referring to. So Fc gamma receptors are the most common type of receptor that interacts with antibodies. It can bind to the Fc domain of many different types of antibodies, these immunoglobulins. So gamma would be for the IgG version. IgAs, IgEs, all the other types of antibodies have their own cognate receptors, but the gamma receptors are specific for the IgGs. And they come in kind of a wide variety. You'll see there in the middle left, Fc gamma R1, on the far left, which is sort of the outlier because it has 3 extracellular Ig-like domains. And then there's 2 A, B, and C, and then there's a third type of receptor, Fc gamma R3A and B. So Fc gamma R3A and R1 are interesting in that they don't have activating, they don't have their own activating domains. So they have to interact with these adapter proteins that have these, what we call ITAMs, or immune, they're tyrosine activator motifs. And so that allows these, once they're bound to IgGs, to signal to the cell to activate and perform some type of activity. So in the case of NK cells, basically an NK cell, which you'll see in green, or sorry, the one on top that's in purple, will be scanning for virally infected cells and would come across one that, say, is infected and has glycoprotein on the surface. And if that is bound to an antibody, then you get sort of a clustering type of motif that is recognized by these receptors. This in turn causes a whole myriad of things to happen, including actin cytoskeletal rearrangement. There's some tethering to the effector or to the target cell, and then lytic granules that contain proteins that would lyse the cell are transferred via microtubules to what we call the immune synapse, or so the synapse between the target cell and the effector cell. Those granules can be delivered directly and then would lead to cell death. And NK cells can go on and what we call serially kill many different times. NK cells do not require antibody or immune complexes for this process to happen. They can also do what's called direct killing, and this would be what happens when it encounters like a cancer cell. In this case, basically activating signals outweigh signals that say, you know, this is a self cell, and that's usually due to sort of some type of stressor or cancer the cell is experiencing, and then the NK cell can go and kill that way as well. So how this process actually happens is not really quite well understood. You know, we know what's needed for this to happen, but the molecular nature is very poorly understood. And this is a detriment to developing therapeutics for viral infection or for developing vaccines that would deliver these types of antibodies. And for that matter, to developing drugs that could also target these same pathways. So my goal is to sort of understand this process a little bit better in a very detailed way, and deliver some paradigms that we can use to design better drugs. But in the case of Fc gamma receptors, one key piece of information that's missing right now is, you know, how do they form these immune complexes that are activating? And similar types of receptors, in the sense that these are membrane bound receptors that need to deliver a signal internally for the cell to do something, are shown at the top. And generally these either are sort of self-activating, they need a ligand, or they form these types of dimeric interfaces that then allow intracellular kinases to bind. And so we're kind of working off this paradigm right now that Fc gamma receptors, in particular Fc gamma R3, in cahoots with its own signaling domain, would form these types of like membrane-bound dimers that you see on the bottom left, that would only form when the antibody is bound to an antigen, and that this, as you can see sort of in the model on the bottom right, would open up these sort of kinase domains to be phosphorylated, and of course, as I've said before, activate the cell. So I wrote this grant last year without any preliminary data, but going off of the structural data that I and others have collected in the past, and sort of the gnomes from immunology, to come up with an idea of trying to figure out if this, you know, basically making a hypothesis and then going out and trying to do exploratory research to look at that. And so just to kind of hone in on what this hypothesis is based off of what I've said, in the case of antibodies that bind in what we're calling ideal presentation, so they're binding in a way that the Fc domains are pointed towards the surveying cell, the NK cell, you're going to get very close interaction of these antigens and the antibodies themselves, which in turn would allow the Fc gamma receptors to bind very closely to each other to form the proper dimeric interface that's necessary, thus opening the kinase domains. And so you would see these early kinases that are associated with ADCC, such as Psych and SAP70, closely associated with these receptors. And then there might be an intermediate stage where the Fc domain is not quite perfectly situated, but you still have some clustering, and you might get some intermediary activation and phosphorylation of the kinase domains. And then there would be sort of the antibodies that we looked at at the beginning that are not activating these NK cells at all, but they're binding to some of the common epitopes. So potentially in those situations, it's just that the Fc domain is pointed away in such a way that it can't really access the Fc gamma receptors, or that it positions them so far apart that the dimeric interface that's necessary for activation is not present. And so I'm using a very novel technique to do this called MinFlux. And MinFlux stands for minimal fluxations, and it's a type of light microscopy technique that was developed very, very recently. And the thing that makes MinFlux unique is that it is able to provide very, very high spatial resolution in the 1 to 3 nanometer range. And this is, if you think about it, a little bit crazy, because with light microscopy, we're using light waves, which are quite large. We can see in the visible spectrum from 4 to 750, somewhere around there. And if we're trying to resolve proteins that are very close together, that's not going to really work. But a Nobel Prize was recently given for some techniques that kind of overcame this barrier, called Palm Storm was one of these techniques, instead was the other. And basically what these technologies did was they came up with some clever chemistry combined with some new ways of shaping the beam that activates these fluorophores on proteins that are labeled to more accurately resolve these very, very close interactions in living cells. So in the case of Palm Storm, which is sort of on the top, basically you have all your proteins labeled with a fluorophore, and they blink. And that stochastic blinking is detected by the microscope and recorded. And this is kind of, they're activated over and over multiple rounds. And over doing that over time, you can sort of get a structure of what you're looking at. Instead, which is below, actually what you're doing is you're first activating beams. So in part 1, you're activating your proteins with a beam that turns all the fluorophores on. Of course, when everything's on, it just looks like a big bright blob. But then you come in with a donut-shaped beam that selectively turns off the fluorophores. And so then you're left after that with very few or hopefully just one fluorophore that's left on, and then you can very accurately determine the location. And so this was revolutionary for the field of light microscopy, because we went from confocal images, which you can see on the top right. In this case, this is the nuclear pore complex, very fuzzy, down to the STED image, where we're getting very close to this nanometer type of resolution. But practically speaking, this does not always give us that super high resolution. Really, the scientists that created this technique were stuck around the 30, 20, and in some cases 10 nanometer resolution, but consistently not able to give us that super crisp image. So what MinFlux did, essentially, in the simplest terms, combined these two technologies together, where we have molecules that are able to be selectively turned on and off, and a donut-shaped beam. And then basically, instead of looking where the molecules are, you're looking where the molecules are not. So thus the minimal fluxations that are recorded at the center of the donut. And basically this ring moves in different directions, and it localizes these proteins based off of the minimum fluxation of light. And so very practically has been able to deliver 1 to 3 nanometer resolutions. As you can see on the right, there's some comparisons to Palm Storm, where, yeah, again, we're sort of in the 10 to 20 nanometer range, and things that are especially close together are not very well resolved at all. And then MinFlux is able to come in and just very accurately produce these beautiful images. So we at Scripps received the very first MinFlux microscope ever, and it's the only one, I believe, currently in the United States. These were developed in Germany, and after the Nobel Prize was given, there was a company that started to develop MinFlux, and now it's a commercially available instrument. So I wrote this whole grant based around the delivery of this microscope at our institute to be able to do some of these really exciting experiments that I've been thinking about doing for a long time. But between the current technologies of light microscopy, their limitations, and then, you know, coming from cryo-EM, obviously we can resolve things at very high resolution, but we're in vitreous ice and we mostly use single particle work. So limitations on both sides of the spectrum, and MinFlux is sort of hopefully going to deliver something that's right in the middle. And so the first part of the experiments that I'm working on is in fixed cells. And so MinFlux is able to basically, you can activate a cell, for example, an NK cell, fix this cell, and then apply dyes through either a tag that has a peptide that can be linked to a fluorophore, or you can use an antibody or a small nanobody. The caveat here is that the accuracy is so high in MinFlux that antibodies themselves are quite large in comparison to the types of localizations that we're looking at. So they're not really ideal. So actually nanobodies are great, or again, like I mentioned, small tags. And as I broke down just a little bit earlier, there's these 3 types of phenotypes that we're kind of looking at. So we'll be developing a panel of antibodies. And then what we're hoping to see is that for antibodies that have really good ADCC activity, that have geometries that are permissive for ADCC, that we get high localization between these early kinases, which would be in one color, A, and then the Fc gamma receptor, which would be in a second color. And then, as a matter of fact, we can actually look at these in 3D. And so we should be able to see on both sides of the cell, the intracellular kinase and the extracellular receptor with really high accuracy. And then these will be some of the first and most highest resolution images of this synapse that we've ever seen in this way. So it should be very exciting. But we're still developing the platform to be able to actually collect this data. What I have been doing on the other side, and something that I haven't really mentioned yet, is that MinFlux, in addition to be able to give us this high spatial resolution, can also look at live cells, and it can track molecules with high accuracy and sub-millisecond accuracy. And so the other thing that I'm really interested in looking at is when these synapses are forming between the NK cell and the target cell, are the receptors moving in a way for ADCC activities that are very good in a more organized way versus those that are not very good in a less organized way. And so again, sort of what I wrote into my grant was what I think this is going to look like. There's no data to support this, but this is just sort of a working hypothesis based off of what we know. But what we're hoping to see is going back to those sort of three types of phenotypes, with the top being good ADCC with close interaction of the receptors, having these Fc gamma receptors moving towards or away from the center of the synapse, in a way that we know already that they do, but are they moving more quickly? Are they moving in a more organized way versus those that are not performing ADCC? We'd expect the receptors to sort of be moving stochastically and not very concertedly. So again, we're hoping to be able to quantify this and provide quantitative data to back up these observations. And the way that I've been going about studying this side of things is to create lipid bilayers. So what you can do in light microscopy, and what's typically done when studying synapses, is to put your target cell and your NK cell together on a slide and allow them to form a synapse, and then look at that synapse. But the problem with that is that NK cells will attack at many different angles, and in order to look at things in a microscope, it needs to be very flat and within a plane that can be seen, within a with a very level Z plane. And so the best way that this has been done in the past is by basically creating like a mock lipid bilayer. So this would be very flat and deposited on the surface of a glass slide, so that you can then plop on your NK cell and it would form a synapse directly within the plane of the microscope. So you can create synthetic layers, or in this case, what I've been doing is taking a cell line that expresses my protein of interest. And right now I'm using Ebola virus as an example, but we'll be going to HIV later. But basically you have a cell expressing your protein of interest on the surface of the cell. We sonicate that and create these nice little lipid vesicles. And those are fantastic because they also contain integrins and other types of activating receptors that might be very important for NK cell activation, but they also contain your antigen of interest. And those can then be assembled on the surface of glass slides to form these really nice bilayers. So we plasma clean them. In this case, I'm actually using these what we call sticky slides. So they have chambers on the top, and you can just stick them onto the glass. The glass is of a quality and a size that's amenable for light microscopy. And then you can assemble the bilayer with all of your antigen present on the surface, and then bind your antibody of interest. And then when you take this into the Minflux microscope, on the left is, it has like a confocal mode. And basically for these early experiments, I've just been taking an antibody that binds to the antigen itself. In this case, it's called 13C6. And that is conjugated to a fluorophore Alexis 647, which is optically works really well with the Minflux system. And we let these, what I wanted to start with was comparing these bilayers to the real system. So in this first case, we just took a cell line that's expressing our protein of interest. These happen to be adherent cells that spread out. And we wanted to see how do these antigens move on the surface of these live cells. So we took live cells, we let them bind to the surface of the glass slide, and then zoomed in. And basically what the Minflux instrument does is it tracks all of these antigens individually. And it can track them for very short periods of time before it starts losing accuracy, and then it goes on to another one. And so all these little squiggles you're seeing on the right are individual antigens on the surface of a live cell moving. The ones, basically it scans from the top to bottom. So you're looking at the first ones that it's scanned in red, and the ones that it scanned later in blue. And if you correlate these with what's happening at the cell surface itself, the ones at the top are sort of the edge of the cell. And so there's a lot more. They're moving more dynamically, a lot faster, which makes sense because that's the leading edge of the cell that's actually moving around, and there's actin kind of probing the surface. And then the ones that are towards the center of the cell, there's fewer of them, and some of them are kind of not really moving much at all. But basically, what we saw was that these antigens are moving dynamically, and some are fast, some are slow, some are stationary. And so we wanted to compare this to the lipid bilayer. Actually, so we wanted to compare this to the native lipid bilayers. So on the left is what I just showed you. And then on the right is the native lipid bilayer surface. And it looks very, very similar. It's a lot more even, which is actually good for our experiments. We see that some antigens are moving very rapidly, and some are kind of stationary. And this kind of is a little bit of a zoom in of what these individual tracks look like. So on the far left is an antigen that's moving very rapidly. And so in yellow is when the Minflex beam started tracking this. And you see sort of linear lines from one point to another. And so that's basically a tracked here, and then it moved to a different space. And it connects those with a line. And then in purple is, actually I got that opposite. Purple is the start and yellow is the end. And so when it's losing accuracy, it will just break off from this antigen and move on to another track. And then in the middle is one that's not moving at all. And so what we think is happening is that these native lipid bilayers have different lipid compositions. So it's thought that cholesterols will kind of slow things down, and they kind of take fluidity away from the lipid bilayer, for example. And so we think that those that aren't really moving maybe are caught in these sort of lipid rafts where there's lots of cholesterol. And then the ones that are moving more rapidly are probably in more fluid areas of the bilayer. And then there's also a bunch of other proteins around that are going to probably get in the way. But basically, that's where we're at right now, which is that we've set up a system that seems to be reflective of the real live cell system, and should be able to deliver us with the results that we're looking for. So that's pretty much all I had on the science side of things. And I know that's sort of rapid fire, but it's early days, and I'm expecting, come talk to me this time next year, we'll have a lot more data to be able to show you, and hopefully some publications out. I can take a quick pause here and answer any questions on the science side of things before I go into the other side of what I wanted to talk about. Or I can just go right into it. So if anyone has any questions, you can just ask them now.
I'll quickly ask a question.
Sure.
And please use the chat, use Q&A, anything you would like if you do have a question. But I'm just a little curious. This is maybe a bit tangential, but all of your schematics and images and planned results are just so refined and so specific and delicate, and I'm wondering how long it took you to prepare all of these kind of expected results and hypotheses and illustrating this, because I think as a grant that doesn't accept any preliminary data, it still seems like creating all of this mock information is still quite an effort.
Yeah, that's a great question. And I think it's a good segue into one of the things I wanted to discuss, which was the grant that funded this money. That funded this research, and where I got the money for it. So I had applied for a K award. So the K award, for those that don't know, is a type of training grant. So it's meant to typically supplement your training as a postdoc. So you need a PhD to get these, with some skill that you need to complete your research. And I was unsuccessful in that twice. And I had, however, a really good relationship with my program officer at the NIH. And all the people in administration that work at the NIH, at least in my experience, have been extremely supportive, and especially young scientists, to help them find ways to get funded. And so I was told there was this new opportunity coming out. And as a matter of fact, it was in January of 2021 was the first time this grant would be available. But they said you should apply for it. And it's called the Stephen I. Katz R01. It is named after the late Dr. Stephen Katz, that was a huge proponent of providing opportunities for early stage investigators to get money to do the research. And so the NIH has, they're very aware of the problem that we're going through currently as scientists, which is that there's more and more scientists, and there's only the same amount of money that's ever been there for the past 10 years or more. So more scientists, same amount of money, maybe even a little less. What do you do? So if you're an early stage investigator like me, you don't have 5 or so years of preliminary data. Typically, you would have good ideas, maybe some early work from your postdoc. You'd apply for a faculty position, and then you'd spend your first 3 or 4 years just collecting data with a startup grant. But what if you can't access that, what we call traditional path? So with the Katz Award, the idea is that for those that have PI status, so you have to have PI status, which you can ask for at your university or your institute, you would then be allowed to apply for this grant. And it has to be a change of direction for your research, and it has to be very new and innovative, and no preliminary data is allowed. So if you include preliminary data, then your application is rejected. So I was like, well, how do you do this? And I love BioRender. And so I thought BioRender is a fantastic platform for especially biologists to create beautiful figures. So I kind of just went through and I sketched out what I thought the data would look like. And then I generated these figures. And so it took me months. And that was a huge part of the grant writing itself, was first creating the figures and then doing the writing. So it did take me a long time. But what else can you do when you don't have preliminary data? And this being even more innovative in the sense that, like, there's not even published similar types of data. There's very little MinFlux data available because it's just a brand new technology and no one really has a scope. So through the use of BioRender and creative sketching and really thinking about it, I was able to create these figures. And I'm really interested to see in say like 5 years when I have the real data, that kind of looking at the before and after of what it's going to look like. So
yeah, I agree.
Long-winded answer.
There's a couple more questions, but I think maybe we'll resume questions at the end, since I definitely want to make sure we can see the rest of your presentation.
Yeah, and I'll say there's not much more to see, but more of me just kind of talking through my background a little bit, and what it's been like to be out and proud gay scientist, and the advantages and disadvantages of being in that kind of space. So I started my research career at the wee age of 14, which for me, I just thought was normal because I'd been interested in science and I wanted to do real good science. But like a lot of middle school and high school students, you don't have access to this type of technology that you need to do some really great stuff. So luckily, I was in a town that had a university, and a friend of mine, both her mom and dad were scientists, and I asked if I could talk to them. And so by doing that, I was allowed to come work in a graduate level laboratory. And I talked with my friend's mother, whose name was Dr. Sabina Heinhorst. And she and I formulated these really cool ideas that led me to kind of my own type of independent research in the lab, where I'd go from, like after school until 7 o'clock every day, and I would spend the weekends there. Um, like in short, I was studying chicken poop, which is, you can ask me about that later, but I did these really cool science fair projects. And that was sort of like going on in the background throughout all of my high school time. And then I stayed on there at USM to go and get my bachelor's degree that I received in 2008. And when I went to college, I joined that lab as sort of an undergraduate helper, and I had already been working in there for 5 years, so I was quite familiar with the environment and was able to hit the ground running and get some publications out. But in the middle of all of that, I ended up coming out as gay, which was very, very difficult because I had spent all of my high school career and at least the first year of being in college, like I never dated, I never went to a gay bar, I didn't drink, I didn't do any of that sort of stuff that a lot of people that surrounded me in Southern Mississippi, had all since experienced. And so it was sort of a curveball for me in the sense that I then was starting to explore being, like someone that could go on dates or someone that could have a relationship, and making new friends, meeting other gay people in my hometown, going to my first gay bar, and suddenly I didn't really have time to do science anymore, and I kind of fell off the bandwagon a little bit. But looking back on it, I was like, well, I think that's fair because I didn't have the opportunity to make the mistakes that I wanted, that other people make in high school. I had to wait until I was in college to do that. So I had to take some time off, but in the middle of all that, I did get to study abroad, which was extremely enlightening for me, coming from a very small Southern town and then going to England, where I studied at Exeter University for 6 months, which is in the southwest part of the island. And it's actually, I would say it's pretty conservative compared to, say like London, which is on the east coast of the island. But I did get to live with 10 other people in a giant flat. I had my own room, and all these people were from all over the world. And I was out and proud for the very first time. I literally had come out to my mom and dad like the day before I got on the plane to go there for 6 months. And luckily, my parents and I have a very good relationship, and it was kind of nice to be able to kind of drop the bomb and then leave, not have to deal with the fallout from that. But over time, they've certainly gotten okay with everything about my personality. But I was able to sort of sow my gay oats a little bit in England, and then come back and graduate. So then I moved on, and rather than going, and again, this is where I guess being a gay scientist, I got a curveball in my life, and that was that most people that I know go from college to graduate school if their intention is to do the journey that I've decided to do. And I wanted to be a professor ever since I started working in a lab back in 1999. But I just didn't feel that it was the right time to go to graduate school. Having just come out a few years earlier, I had never really been in a relationship. I had never really lived outside of my hometown, except for, being in England for a few months. So I decided to be a technician, and I moved to Atlanta, which we call the gay capital of the South. And friends of mine in Mississippi would journey there usually once a month. It's like a 5-hour drive from Hattiesburg, and stay with friends. And we all decided to move there together in 2008 after we all graduated college. And those were some of the best years of my life because I was able to do science. I was able to do other things. I taught color guard and a local marching band, and I was able to work in some restaurants part-time, which I loved doing. Really just sort of like live my life, and then realize at the end of that journey that I still really loved science, and that was the route that I wanted to go. So I applied all over and visited this little place called La Jolla, and fell in love first with the beautiful weather here and the fact that my potential graduate school would be on a mesa that overlooked the Pacific Ocean. I just thought, this can't get much better. But in particular, I loved the fact that Scripps, and this is still true today, was a place where the focus is very much centered on the basic research, the science itself, with the caveat of we're obviously very interested in human health, and in particular, like neuroscience and infectious disease. But I thought this would be a great way to caveat into science that would be more impactful, more immediately impactful. And so I've been at Scripps ever since. And I came to Scripps with the, yes, I am gay. I put that on all my applications, and I've just always made a point of being very straightforward about my sexuality, because there's many different reasons. Like in some ways, I felt it's the only type of diversity card I can really play. But really, at the heart of it, it's also been something that has shaped my pathway, as you can see, very, very clearly shaped who I am, and in many ways, the type of science that I choose to do, and gotten me to where I am today. So that's just been sort of very important to me. And being in a city that embraced that type of personality has been pivotal to my success here. So I joined a lab, and I was co-advised by two people. And then I stayed on in that lab. This is Dr. Andrew Ward as a postdoc, and ended up meeting my husband, and my advisor, Dr. Ward, came to our wedding, and he's been to some of my crazy drag fundraisers, which I can tell you about if you're interested. But has always been fervently in support of diversity in not only his lab, but allowing people to be themselves and be open about who they are. And I think that that's been also good in the sense that I've been approached by administration at Scripps, and I've been able to stay on and be more involved in diversity initiatives, especially in more recent years, because people know that I'm a guiding light, in the sense that I'm easy to talk to. I love to talk to people, and I kind of market myself as a conduit between, gay Scripps and the rest of San Diego, and maybe people that aren't quite comfortable approaching others. So just trying to be like a voice that is a synapse between the administration and the actual community that we live in. So in that spirit, I have been involved with our DEI committee at Scripps recently. And coming from a science fair background, as I mentioned, I'm a huge proponent of science fairs, like the best way to get young, diverse scientists into research, because that's what got me hooked, was doing science fair and being successful at that. And so we started I helped start a award for the local San Diego Science Fair called the Scripps Young Scientist Award. And we're about to present some more this year. This will be our third or fourth year presenting, and I hope that continues on, and that maybe some of those scientists that we awarded will someday go into research themselves and maybe come to Scripps, which would be great. And then the other thing that I'm heavily involved in is the San Diego American Flag Football League. And so it's a recreational league that's been around for 18 or so years now. And basically we try to be a platform for gay people and men and women alike, everywhere in between, as well as allies, to come together and play flag football, and then also have an opportunity to socialize with other people. So after all of our games, every Saturday from March until June, we go to a different bar or restaurant in Hillcrest and have a social. And then we have all these great activities that we do. This weekend, we're doing karaoke at Red Wing, which is on 30th and North Park, and it's always super fun. And then 2 weekends ago, we did Laces and Lashes, which is a jocks and drag kind of contest that we started, and has just blossomed into this huge thing. And it's like a big pageant, and it's taken very, very seriously. There'll be a video up of that on our YouTube channel in the next few weeks. And we raise money for the center, which is a crisis center in Hillcrest. And also all of this money goes to our operating funds. And so I'm poised to kind of lead that organization, and all of these skills that I gained from working even outside of science have kind of contributed to me being successful as a PI, and hopefully continuing to be successful as I move forward. Yeah, I guess that's all I wanted to say, except that I talked to our administration about starting Quest up again. So I don't know if anybody knows what that is, but Evan, I will certainly be talking to you about this soon. But Quest was something that was a cross Mesa, like biannual mixer that I think UCSD sort of put on, but all of the institutes in the area contributed to that. And it stands for queer engineers, scientists, and maybe something with technology. But we're thinking about starting that up again as sort of a catalyst for getting an LGBTQ organization at Scripps, which we don't currently have. We have a graduate sort of led diversity initiative, and we have an INWIS chapter, which is for women in science, but we don't really have like a gay organization. And so we've always lived vicariously through our sister organizations in the area. But we're hoping that by hosting Quest and starting that up again, that we can show the administration that there's a lot of support. So the idea is to get a ton of people to come, probably sometime over the summer, and we'd have it catered, and we would pay for everything in the beginning. But I just wanted everyone that's listening to be aware of that, and that it's coming soon enough. So that's all I had to say. And if there's any questions, I can answer those couple that came up before, or if there's any new ones.